My name is Arshad. I'm from Pakistan and I'm working in Brazil in the biophysics department. And my presentation is about the nanosystems formed by amphiphilic antimony complexes incorporating amphotericin B for the treatment of leishmaniasis. And the outlines include the introduction, what is leishmaniasis, and the geographical distribution of the disease, the current chemotherapeutics, the amphotericin B, what is amphotericin B, and objective of the study, results and discussion and conclusion. Leishmaniasis is a significant neglected tropical disease. It's a group of diseases caused by the protozoan parasites of the genus Leishmania. And the infection is transmitted by the bite of sandfly of the genus Phlebotomine and Lutzomia. There are two morphological forms of the parasites, amastigotes and paromastigotes. Life cycle. In the life cycle, the sandfly takes blood meal and takes the macrophages infected with the parasites. And inside the midgut of the sandfly, the paromastigote converts to paromastigote form, then to the infectious metacyclic form, and okay. So when this infected sandfly bites mammalian host, it injects the parasites inside the human body, where it is taken up by the macrophages. Inside the macrophages, they are converted to the amastigote form, and in this way the life cycle continues. There are three types of leishmaniasis: visceral leishmaniasis, cutaneous and mucocutaneous leishmaniasis caused by different strains of the leishmanias. And the most fatal case of the leishmaniasis is visceral leishmaniasis. And they have different symptoms like fever and substantial waist loss, etc. Risk and geographical distribution of the disease. So it is endemic in 98 countries. There are 350 million people are at risk at this time to the disease, and 1.5 to 2 million cases occur each year. And there are 12 million people are affected by leishmaniasis. About 60,000 people die annually because of the disease, and this disease is prevalent in all the 5 continents of the world, and most in Bangladesh, Brazil, and India. The current therapeutics for the leishmaniasis are the pentavalent antimonials, and which includes glucantime and pentostam, which are the first-line drugs in developing countries for the cutaneous leishmaniasis. The mechanism of action of these drugs is still not known, but there are some proposed mechanisms in which the antimony interact with DNA and cause apoptosis of the cell. But there are disadvantages of these drugs, for example, cytotoxicity and resistance and high cost. What is amphotericin B? This is my main objective of my work. Amphotericin B is a standard antifungal drug, which we also use for the treatment of leishmaniasis. In case where there other drugs do not work, so we use the amphotericin B, but this is highly toxic in aggregated form in the solution. To reduce its cytotoxicity, various strategies have been used to incorporate the amphotericin B in the nanocarriers. For example, nanoemulsions, micelles, and liposomes. Objective of the current research to investigate, to seek for the new approaches to treat the leishmaniasis, to synthesize new drugs or improve the existing, which are safer with minimum side effects, which are cost effective and orally administrable. Orally administrable just to avoid time and muscular pain. Specific objective of this project is the incorporation of amphotericin B in the nanosystem that we synthesized, which are based on the glucamide series. It is a surfactant, and so we use 2 surfactants, that is with 8 carbon chain and 10 carbon chain, and we call it, in this presentation, I will call it L8 and L10 because of the number of carbons. Then we studied the kinetics and the stability of the new formulations of the amphotericin-loaded nanosystems, and the evaluation of these new formulation as anti-leishmanial drug in vivo. So here I will present just a little about the preparation, how we prepare. So we mix the antimony with the surfactants, and the process is there, but this is not very important to know. And this is the nanosystem, which has a size from 100 nanometer to 200 nanometer, and this is confirmed by mass spectrometry and NMR spectrometry. So analysis of amphotericin B content and drug encapsulation efficiency by an HPLC based technique. Preparation, how we prepare this nanosystem, these formulations, was by mixing of amphotericin B with the nanosystem, and the encapsulation efficiency was evaluated by using reverse phase HPLC techniques, which is a standardized techniques, by the following formula, the quantification of the amphotericin before filtration and after filtration. So what we found by the HPLC technique is that 84% of the amphotericin B gets incorporated into the 8-carbon chain nanosystem and 83% into the 10-carbon nanosystem. So as I said, that amphotericin B is cytotoxic in the aggregated form. So here in the nanosystem, we try to incorporate the amphotericin B in a monomeric form, and which we confirmed by UVVs, ultraviolet spectra, absorption spectra, in which we can see that this peak, about 300, corresponds to the aggregation state of the amphotericin B, which we see that this is for the free amphotericin B in PBS, which has a pH of 7.2. And when we mix this amphotericin B with the nanosystem, so they lose the aggregation state and they incorporate in the nanosystem in the monomeric form. Here we have the same results in the HCl, because at physiological pH and HCl at the pH in the stomach that we need to maintain the stability of the nanosystem of the formulations. This is a circular dichroism spectra of the amphotericin B, where it shows the similar results in which the amphotericin B is in aggregated form, in free form, but when we incorporate this amphotericin B in the nanosystem, it converted to monomeric form, which is not cytotoxic. And with the 8-carbon chain nanosystem, we have the similar results in which the red line, the red peaks, corresponds to the aggregated form of the amphotericin B, and in the other peaks, you can see that this peak is disappeared, which means that the amphotericin B is in the monomeric form. This study was done to see the stability of the formulation in different solutions with DPH probe. DPH probe demonstrate about the hydrophobic environment of the nanosystems. So here we can see that it is very stable in water during a time from 1.5 hours. Okay, and the similar results were obtained for the 10-carbon nanosystem, in which we can see that the formulation is showing a stability by the DPH probe in water and BPS solution. Infection and treatment protocol of the mice infected with Leishmania amazonensis and Leishmania infantum. It doesn't have too much biotechnology, but it's okay, right? It works. So, first of all, we infect the mice in day 1, and then after 7 days we start the treatment with our formulations that we synthesized. And the 28th or the 65th day, we extract the Leishmania from the spleen and the liver and did the PCR. So here I will show just the PCR results. I don't have too much results because this is just the start of my project. So in the PCR results for the live parasites inside the spleen, we have analyzed by PCR the suppression of the parasites in the percentile. So only our formulation gave a significant difference to the control. This is our control, the amphotericin B by intraperitoneal way. The other drugs also show the significant results, but because of the high standard deviation, this is by the software they are not showing a significant. And in the cutaneous leishmaniasis, we can see the same SBL10 amphotericin B administered orally is showing the significant results. And this is just a start of a project. So I have just these results that I have presented. And in the conclusions, amphotericin B is highly admissible in the monomeric form in the SBL8 and SBL10 dispersions. SBL8 amphotericin B and SBL10 amphotericin B formulations are stable in solution upon dilution at neutral pH as well as acidic pH. SBL10 amphotericin B is less cytotoxic than SBL8 amphotericin formulation in vivo. This study established for the first time for the potential of mixed SBL10 amphotericin B formulation for the oral treatment of both cutaneous and visceral leishmaniasis, indicating their potential for further development and applications. And here my presentation comes to an end.